So, protein person. What is the structure of proteins? What are they made of?
Amino acids.
Amino acids. So we have amino acids. How are they put together?
They're wrapped up into a polypeptide.
What's the bond that secures different amino acids together? A peptide bond. Okay, so peptide bond, listen please, secures amino acids together. Once you have a bunch of those all together, now they're called a polypeptide. See, look. Peptide, peptide. Peptide bonds. Many peptide bonds will form a polypeptide. Poly means more, like lots, right? Excuse me. Poly means a lot, so we have a lot of peptide bonds, okay? Then that polypeptide is wrapped up into a protein, right? Yeah, it's all packaged up, okay? Okay, categories to the proteins. What categories were there for proteins?
It wasn't really.
So what do we say about proteins' characters, sir?
We said their qualities are determined by the types of amino acids.
Qualities are determined by amino acids and the shape that they're put together. Good, okay. Read, what's the major function of proteins?
They form the structural parts of the cell and act as antibodies or enzymes.
So cell structures, enzymes, and things like muscle in our body are primarily protein, right? Yeah. Good. What's an example of a protein? Dylan, what's an example?
Where was that?
The first page.
I don't know.
Last question.
Could you tell us what it is? I never got an example.
Just read what's on your paper.
Okay, that group. Matt, can you share?
Hemoglobin is a quaternary protein that contains 4 individual polypeptide chains that combine to form functional hemoglobin.
Yes.
Oh,
All you had to say was hemoglobin.
I thought that you meant like there was gonna be like a potato or something, one word, not like a big old.
It is one word.
A potato?
I get it now.
Okay. So hemoglobin, yeah, is a type of protein, and it has an iron component to it, which is the heme. The heme is the iron. That's why your blood turns red when it's exposed to oxygen, because it comes oxidized. It's chemistry. Okay, anyways. There's a heme group, yeah, in the globin, and it really likes oxygen. That's why your blood can transport oxygen, because your blood is made of red blood cells, which are made of hemoglobin, which transport oxygen. Whoa, so cool. Hey, also, by the way, what is the thing that transports these proteins from our guts to our muscles?
Intestine.
Okay, once it gets to your intestines, how does it get from your intestines to something like your muscle? Capillaries, which inside the capillaries is a fluid known as blood. Okay? Yeah, I know you guys said it too, but then I was hearing lots of other things, so I just wanted to make sure we all wrapped it up. But yes, the capillaries are on the intestines, and in the capillaries is blood. So the capillaries absorb the proteins or the carbohydrates or the lipids and transport them in the blood. That's like a super key, important concept to know throughout the whole Bio 20 of this unit. Blood transports nutrients. Okay? All right. Next. Okay, let's talk about carbs. What are carbs made of, Addison?
Sugar.
Sugars. Do we need to know anything more? No. Okay. So your textbook has a lot of pictures of sugars. Just don't worry about those. We will do those in Chemistry 30. Just know that carbohydrates are made of sugar chains, let's say specifically, because we have to have lots of sugars all together. Once you have a lot of sugars all together, that makes a carbohydrate. Are there types of carbohydrates? What are they, Babian?
Monosaccharide.
Okay, monosaccharide. So saccharide means sugar. Okay, this is like a term for sugar. So this means one sugar unit. So it's like one sugar units are all put together in a chain. Okay, so one sugars in a chain. And then what else? Disaccharides. So how many sugar components are in this chain? 2. So we have lots of 2 sugar components all put together. And then there's polysaccharide. So many sugars in a chain. Polysaccharide. Yeah. Perfect. Okay. What's the major function of carbohydrates in our bodies, Hannah? Yes. Energy. Which process uses the broken down carbohydrates to make energy? Yes. Cellular respiration. You guys are the smartest grade 11s I teach.
We're the only ones.
See, I told you you're smart. Okay, so what's an example? Lane, tell me an example of a carbohydrate
Potato.
that you like to eat at A&W.
Ooh.
It's fried and greasy, and you dip it in ketchup.
Chicken fingers.
Oh, okay.
Hamburgers and ketchup. Okay, fries.
Fries, okay.
I
So,
like A&W fries.
so like a potato.
I actually hate A&W fries too. Actually, I like them.
Potatoes are just starch, right? Potatoes are carbohydrates. When we break them down, now they're sugars. What are they coated in? What are these French fries at A&W coated in? Oil, which is a lipid. Okay, so now we're on to lipids. So, what is a lipid made of, Jess?
Glycerol and fatty acids.
So glycerol, and we could say a glycerol backbone, meaning like it's kind of the framework. So it's like here's a glycerol, and then what comes out of the glycerols? What are those? Oh, you already told me, fatty acids. Now, they could be either triglycerides, which have 3 fatty acid chains, or phospholipids, which have 2. But just know that it's the fatty acids and the glycerol backbone. That's key. Good. Okay, categories. Evan.
Fat, oil, and wax.
Fats, oil, and wax. So how do we distinguish these? Well, fats are solid at room temperature. Oil are liquid at room temperature. You guys know that. Fats, think of like butter, right? Oil, olive oil, right? One's solid, one's liquid at room temp. What about waxes? What's special about those, Evan?
It's a long chain. Long chain.
Long chain lipid. So is it longer than the fats and the oils? What makes it special? I actually don't know.
It says it's made of fatty acids and alcohol or carbon rings.
An alcohol carbon ring?
It says or alcohol or carbon ring.
So there's some chemical property that makes it a little bit different. We don't need to know about it. Well, in Chem 30 we could know about it, but not right now. Okay. Describe the major function of lipids in our bodies. Who haven't I picked on? Has everyone been picked on? What are they screaming about?
Ebola.
It's coming.
Okay, so honest question. You have to like meet somebody with Ebola to get it, right? Like you
Yes.
Okay, so I don't think I should have Ebola.
No, there's no confirmed reported cases of Ebola in Canada yet. Confirmed by the Canadian Health.
Boom.
There are rumors of Ebola.
In Edmonton?
Yes.
In Grand Prairie?
But if they were actually reported cases, we would know for sure because Alberta Health would be emailing every single school. You know? So it's rumors. Not that that doesn't mean that it's not true. I don't know if it is or not. But if it's not been a declared confirmed case. So just always act with caution, but don't freak out. Okay. Freaking out doesn't do anything for you rather than spend your mental energy. So okay. What's the purpose of lipids in our bodies? Matthew knows.
What's the purpose for?
Yeah.
Store energy components, the cell membranes, act as a cushion for organs, carry for vitamins A, D, and K.
Store energy cushion, cell membrane. Yeah, that's a really important one. All of our cell membranes are made of phospholipids. This is kind of one of those details that we learned in Science 10, so you should definitely remember that one. That's like a memorization point. Good. Okay. Good, so those are our macromolecules. Now here's just a couple things. We already talked about monosaccharide and poly. So these are all about our carbs, and we talked about them. So what's dehydration, synthesis, and hydrolysis? Let's do dehydration synthesis first. Hannah, what's dehydration synthesis?
Formation of larger molecules from smaller molecules.
Good.
During water.
Formation of larger molecules from smaller. And what did you say about water?
Thicker than water.
Yeah. And removes H2O. If you forget this, just look at the word dehydration. When we think of dehydration, you have less water, right? So water is removed. Synthesis, we're making something. You have to make something from something else. So we're making something big from something smaller and taking out water. Okay? So break the word down a little bit for yourself so that you don't have to do lots of memorization. Okay, hydrolysis. Okay, so what do you think if these are kind of different from each other? Hydro, what are we doing with the water? Are we taking it away or adding water? And what else happens, Addison?
Larger molecules get broken down into smaller.
Yeah, so these are opposite processes. So we add water to break down bigger molecules. So like I said again, don't make it harder than it is. Look at the word and think about what does this word mean? Hydro. I'm putting water in. If this is the opposite of this, you're breaking down the bigger molecules into smaller ones. Saturated fatty acid and unsaturated fatty acid. Okay, we do this in detail in chemistry, but what I need you to know about saturated fatty acid is it has every carbon has full amount of hydrogens. An unsaturated fatty acid is the opposite. At least one double bonded carbon in molecule. Now, basically, it's like, why is this good or bad? Well, a saturated fatty acid has so much hydrogens on it, and without getting into the chemistry details of it, it's like it has these prongs, okay? Saturated fatty acids have a bunch of prongs. When they get touching each other, they all connect. Okay? They connect and they're stuck to each other. So if you have a lot of saturated fatty acids, when your blood is trying to transport these lipids, they get stuck and clog your arteries. Okay? So saturated fatty acids are harder for your body to process and transport, and then you get clogged arteries. This is important to know. If you did not write that down, so need to know why it's unhealthy or less healthy to have lots of it, right? Because it's harder for your body to process and break it down, so they build up and it causes blockages in your arteries. Okay? That's like a test question. You need to know why saturated fatty acids aren't as healthy. It's in your textbook, a whole explanation about it too. So unsaturated fatty acids are the opposite then. They're easier for your body to break down. So if we think about it, a saturated fatty acid is something like butter. Generally it's our fats. Olive oil is easier. It's unsaturated. Okay? So generally when we think of healthier options, it's usually the oils that are unsaturated and the heavier, thicker ones that are solid at room temperature are less healthy. That doesn't mean that butter is unhealthy for you. It means that a lot of butter is unhealthy for you. Just as if you drank a thing of olive oil, that's unhealthy too, right? Like everything in moderation, right? It's just that comparatively, a lot of butter can do quicker harm to you than the same amount of oils or an unsaturated oil. So it's everything in moderation. Don't go home and say, Mom, we cannot eat butter anymore. No, that's not what we're saying, right? It's everything is in moderation, okay? Things like, that's why you go easy on the bacon. Because bacon, fat, is a very saturated fatty acid. When you cook it, it turns into the liquid, right? But then you still eat it, right? Before it turns back. Yes, so tasty. But too much bacon clogged arteries. That's why, saturated fatty acids.
So how much bacon do you have to eat before your arteries clog?
Well, I don't know, Dylan. It depends how much exercise you do, how strong your heart is, how much your your metabolism. It depends on all those things. Shape of phospholipids. What's this thing? What's the head on the top of a phospholipid? Where's the glycerol in this? Pardon? No, those are the fatty acids. What does your textbook have? So page 247. So you have the phosphate group. There would be like a glycerol backbone right here. And our fatty acids. Denaturation and coagulation. So this is in reference to our proteins. So when we denature something, the process when the bonds of a protein molecule are disrupted, causing temporary change in shape. So this is change in shape, temporary change in shape. That's the biggest thing, is that it's temporary. Coagulation means what? It's permanent. So when you cook an egg is pretty much just all protein, except the yolk is what? The fat. The yolk is the fat. The white stuff around, that's the protein. So when you put when you put an egg in the frying pan, the white bubbles up and it changes color, right? Well, it's clear, obviously, at first, but then it changes to white. That's because those proteins have coagulated, and they've permanently changed shape and become that white texture that we have of a cooked egg. Because you can't uncook an egg, right? You can't. You can't uncook an egg.
You're really making me want eggs here.
Oh, sorry, and bacon. So you can't uncook an egg. That's why you think about, okay, that's coagulated. You can't change it. Denaturing is just a temporary change. So that's really important that you know these. So even though this was kind of a we teach each other stuff, this is still super legit, important testing information. I will give you a study worksheet that will help you kind of zone in on what were the most important things as well, okay?